Welcome back to the deep dive. I've always been fascinated by space exploration, but it's easy to forget the incredible challenges our bodies face outside of Earth's atmosphere. Today, we're diving into one of those challenges specifically for you. You wanted to understand the impact of microgravity on human muscles and how that affects astronauts.
It's remarkable how adaptable the human body is. But even with rigorous training, microgravity presents a unique set of obstacles.
Absolutely. And to unravel these mysteries, we'll be using this fascinating scientific publication, Advancing Insights into Microgravity-Induced Muscle Changes, using Caenorhabditis elegans as a model organism. We've also got diagrams of muscle structure, illustrations of cellular changes, and even information on how scientists mimic microgravity right here on Earth. But first, let's break down what microgravity actually is and why it causes so many issues.
Well, in simple terms, microgravity refers to the near weightlessness experienced in space. It's not the absence of gravity, but rather a significant reduction in its apparent effect.
So it's not that gravity disappears, it's more like everything is in constant free fall. That must have some pretty wild effects on the body, beyond just making you float around.
Precisely. Think about it on Earth. Our muscles constantly work against gravity to maintain posture and movement. In space, that resistance is practically gone. As a result, even with exercise and a carefully planned diet, astronauts can lose significant muscle mass, sometimes up to 20% during extended missions.
Wow, 20%. That's a huge amount, even with countermeasures in place. It makes you wonder what's happening at the cellular level to cause such dramatic muscle loss.
That's the million-dollar question. If we can understand the specific mechanisms behind muscle atrophy and microgravity, we can develop better strategies to protect astronauts during long-duration spaceflight.
That makes sense. It feels like we need to zoom in almost microscopically to understand the root of the problem.
Exactly. To do that, we need to delve into the fascinating world of cellular biology. Muscle atrophy, in essence, is like cellular downsizing. The muscle cells shrink and lose volume. And a key player in this process is a complex system called the ubiquitin-proteasome system, or UPS for short.
Okay, ubiquitin-proteasome system. That's a mouthful. Break that down for me a little. What is the UPS, and how does it contribute to muscle loss?
Imagine the UPS as a cellular recycling center. It's responsible for breaking down proteins that are damaged or no longer needed, clearing the way for new ones. But during muscle atrophy, the system goes into overdrive.
So instead of just routine maintenance, it starts demolishing things more aggressively.
Precisely. Specific molecules within the UPS, let's call them tagging molecules, mark muscle proteins for destruction. The more active these taggers become, the faster muscle proteins are broken down, leading to the shrinkage we see in muscle atrophy. And what's particularly intriguing is that microgravity seems to trigger this overactivity in the UPS.
You're essentially saying that in microgravity, this cellular recycling system gets a bit trigger-happy and starts breaking down muscle proteins faster than they can be rebuilt. That explains the significant muscle loss astronauts experience even with exercise. That's a pretty huge discovery, isn't it?
It is. This understanding has opened up new avenues for research. If we can find ways to regulate the activity of this system, perhaps through specific medications, we might be able to prevent or reduce muscle loss in space.
Now, that's what I call a game changer. But how do scientists even track these changes in the first place? Do they have tiny cameras inside the cells?
Not quite, but they do have something called biomarkers, which are measurable indicators of what's happening within the body. One biomarker crucial for muscle health is a complex called the dystrophin-associated glycoprotein complex, or DGC. It's like a bodyguard for muscle cells maintaining their structural integrity and sensing mechanical stress.
So if the DGC is compromised, it's a sign that muscle health is in jeopardy. I'm guessing this is where those diagrams of muscle structure come in handy.
Absolutely. By studying the DGC in both humans and a surprising research subject, a tiny worm called C. elegans, scientists can glean a clearer picture of what happens to muscles at a microscopic level during spaceflight.
Wait, a worm? Tell me more about this C. elegans astronaut.
C. elegans is a fascinating creature. Despite its simplicity, it shares many fundamental biological pathways, with humans making it a powerful model organism for research. Plus, its small-sized transparency and short lifespan make it ideal for space studies.
So it's like having a window into the cellular processes happening in a much simpler organism. That might translate to humans. That's a pretty clever way to study these complex changes.
It is. But let's shift our focus back to the human experience for a moment. What have scientists actually observed in astronauts who've spent time in microgravity?
That's a great point. It's easy to get lost in the cellular details, but ultimately it's about understanding the real-world impact on those brave space travelers. What have they found?
Well, studies have shown significant reductions in muscle fiber diameter, sometimes by as much as 19%. Additionally, there's evidence of myofibril width reduction and Z-band length decrease, all pointing to substantial structural changes within muscle tissue.
So microgravity essentially weakens the very building blocks of muscle. And it sounds like it's not just about size, it's also about the structure of the muscle itself. What are the implications for astronauts' performance?
Precisely. This structural weakening means reduced strength and endurance. Imagine trying to perform complex tasks in a demanding environment with compromised muscle function. It highlights the critical need for effective countermeasures.
It's fascinating how something as seemingly simple as gravity can have such a profound impact on our bodies. But we've talked a lot about muscle structure and breakdown. What about the power source of these cells, the mitochondria? How does microgravity affect them?
The mitochondria, the powerhouses of the cell. It turns out that microgravity throws a wrench into their operations as well.
So instead of a cellular power grid, it's more like a blackout. What are the consequences of this mitochondrial mayhem?
It's not a complete blackout, but more like a brownout. Studies show that genes involved in mitochondrial energy production are downregulated in microgravity, meaning their activity is reduced. This leads to a decrease in ATP production, the primary energy currency of cells.
So the cells are essentially running low on fuel. What does that mean for their overall function?
Imagine trying to run a marathon with limited energy reserves. It's not sustainable. This energy depletion affects a multitude of cellular processes, potentially impacting everything from muscle repair to overall metabolism.
And to add insult to injury, we haven't even talked about those reactive oxygen species, or ROS, which we mentioned earlier. I have a feeling they're not helping the situation.
You're right, they're not. ROS are a natural byproduct of cellular metabolism, but under stress, like in microgravity, their production increases, leading to oxidative stress, which is like rust forming inside the cell, causing damage to important cellular components.
It seems like microgravity is creating a perfect storm inside the cell. It disrupts muscle structure, throws off energy production, and then bombards the cell with these harmful molecules. It's no wonder astronauts face such challenges. But you mentioned earlier that scientists can simulate microgravity here on Earth. It sounds like something out of a science fiction movie.
It does, doesn't it? But these simulations are crucial for studying microgravity's effects in a more controlled environment. They can use specialized devices like clinostats, which rotate samples to average out the effects of gravity, or even powerful magnets to create a force that counteracts gravity.
So they're creating mini environments that mimic space right here on Earth. That's amazing. And they can use these environments to study those C. elegans astronauts we talked about earlier.
Precisely. By observing how C. elegans respond to simulated microgravity, scientists can gain valuable insights into the cellular and genetic changes that occur in a space-like environment.
So by studying these worms, we can learn more about how human muscles might react in space. It's like having tiny, transparent astronauts revealing the secrets of microgravity. It's incredible.
It is. And remember that table from the research paper you mentioned? Table 2. It contains a wealth of information about specific genetic and protein changes observed in C. elegans muscle under microgravity. It really dives deep into the molecular nuts and bolts of what's happening inside these cells.
Table 2 it is. We've definitely got to unpack that in our next part. It sounds like we're just scratching the surface of this fascinating research.
We are. There's so much more to explore.
Join us in part 2, where we'll delve into those genetic changes and uncover even more insights from our tiny worm astronauts. You won't want to miss it. Welcome back to the deep dive. Last time we were left hanging with the promise of diving into Table 2 from the C. elegans research. It sounds like it holds some pretty specific details about how muscle is affected at a genetic level by microgravity.
It does. Table 2 really gives us a close-up look at the molecular machinery that's impacted by the lack of gravity. One of the key findings is the significant downregulation of the myo3 gene in the worms exposed to microgravity.
Okay, myo3. I remember you saying it's a gene, but refresh my memory on what it actually does.
Think of myo3 as a blueprint for a crucial protein called myosin heavy chain A. This protein is like the engine that powers muscle contraction. If myo3 is downregulated, it means less of this essential protein is being produced.
So less myo3 means weaker muscle contractions. That seems pretty straightforward. But how much of a difference are we talking about here?
The study actually found a 73% decrease in myo3 expression in C. elegans exposed to microgravity. That's a significant drop. It really underscores the impact that microgravity can have on muscle function at a very fundamental level.
Wow, 73%. That's a staggering difference. No wonder these changes are so noticeable in astronauts. But is MYO3 the only gene affected? There must be others involved in this complex process.
You're absolutely right. There are a whole host of genes involved, and the research uncovered changes in several others. For example, they found downregulation of the UNCFF4 gene, which encodes a protein called paramyosin.
Paramycin, another protein involved in muscle function. What does this one do?
Paramyosin plays a critical role in regulating muscle contraction and maintaining the structural integrity of muscle fibers. So it's not just about the power of contraction. It's also about control and stability within the muscle.
It's starting to paint a clearer picture. Microgravity isn't just making muscles weaker, it's affecting their ability to function properly at a very basic level. Are there any other noteworthy genetic changes mentioned in Table 2?
There is one more I think you'll find particularly interesting. The study also found reduced expression of genes involved in the worm's pharyngeal muscle function. Genes like PEB1, SAE22, and FAE4.
The pharynx. Now that's a new one. Isn't the pharynx part of the digestive system?
It is. The pharynx is essentially the worm's throat. It's responsible for sucking in food.
So these findings suggest that microgravity could even affect something as basic as the worm's ability to eat.
It's a fascinating observation, and it highlights how widespread the effects of microgravity can be. It's not just about skeletal muscles. It can impact various systems within the body.
It makes you wonder if similar changes might occur in astronauts' digestive systems during prolonged spaceflight.
It's certainly an area that warrants further investigation.
Yeah.
And remember, we're seeing all these changes in C. elegans, a relatively simple organism. The complexity magnifies significantly when we consider the human body.
This is where those parallels between worms and humans become really important, right? What we learn from C. elegans could provide valuable clues about what might be happening in astronauts.
Precisely. While there are obvious differences between worms and humans, the fundamental biological processes are remarkably similar. So studying these genes in C. elegans gives us a window into the molecular mechanisms that could be contributing to muscle atrophy in humans during spaceflight.
It's almost like having a simplified model to test and observe these changes in a more controlled environment.
Exactly. And that's why research using C. elegans and other model organisms is so crucial. It allows us to explore the complexities of human biology in a more manageable and ethical way.
It's amazing how these tiny worms are helping us unlock the secrets of space travel. I'm curious, how do they actually study these C. elegans astronauts? Do they have miniature spacesuits?
It's not quite that elaborate, but it's still pretty impressive. They've actually sent C. elegans to the International Space Station. They travel in specially designed containers that provide a suitable environment, ensuring they have the necessary nutrients and protection.
So these worms are true space travelers.
Yeah.
But I also remember you mentioning ways to simulate microgravity back here on Earth. How do they use those techniques to specifically study muscle atrophy?
Those simulated microgravity environments are incredibly valuable for research. They allow scientists to conduct experiments with greater control and flexibility. One method involves using those rotating devices we discussed earlier, like clinostats, to create a constantly changing gravitational field that mimics the effects of weightlessness.
So they're essentially giving the worms a taste of space without sending them into orbit. Clever. And I'm guessing they compare these space worms to their counterparts here on Earth to isolate the effects of microgravity.
Exactly. They can then analyze changes in muscle structure, gene expression, and protein levels to pinpoint the specific effects of microgravity.
And what kind of insights have they gleaned from these simulated space adventures?
These studies have revealed some fascinating findings. For example, C. elegans, exposed to simulated microgravity experience, increased oxidative stress, similar to what we discussed earlier, and they also activate cellular repair pathways in an attempt to combat this damage.
It sounds like those reactive oxygen species are causing trouble even in these simulated environments.
They are. And another intriguing observation is that simulated microgravity can affect the worm's reproductive capabilities, just like an actual spaceflight. It's another example of how these studies can mirror the effects seen in real space environments.
It's mind-boggling how something as seemingly simple as changing the gravitational field can have such a ripple effect on various biological processes.
It really highlights the intricate connections within biological systems and how sensitive they are to changes in their environment.
We've covered so much ground today, from the cellular breakdown of muscle to the genetic changes observed in C. elegans, and even touched on the ethical considerations of using model organisms in research.
It's been a deep dive in every sense of the word. But we've only just begun to explore the broader implications of this research.
You're right. What does all of this mean for the future of space travel and even for human health here on Earth? I'm ready to explore those big questions.
I'm excited to delve into that as well. Join us in part 3, where we'll take our understanding of microgravity and muscle atrophy and apply it to the bigger picture. It's going to be fascinating.
Welcome back to the deep dive. We've journeyed from the intricacies of muscle cells to the fascinating world of C. elegans astronauts, all in pursuit of understanding how microgravity wreaks havoc on our muscles. Now, we need to shift gears a bit and think bigger picture. What does all this research mean for the future of space exploration? And are there any surprising takeaways for us Earthbound folks?
It's a crucial question, isn't it? As we set our sights on longer missions and even consider establishing permanent outposts on the Moon or Mars, safeguarding the health of astronauts becomes paramount. The implications of prolonged muscle loss go far beyond discomfort. It could compromise mission success and even endanger lives.
It's a sobering thought, especially considering how difficult it is to combat muscle atrophy, even with countermeasures like exercise and diet. It feels like we need a more comprehensive approach, something that addresses the problem at its root.
I completely agree. The research we've been discussing, particularly those insights into the UPS system and the genetic changes observed in C. elegans, opens up exciting possibilities for developing targeted therapies. Imagine medications that could regulate the breakdown of muscle proteins or even stimulate muscle growth in microgravity.
That's a fascinating prospect. It feels like we're on the cusp of a new era in space medicine, where we move beyond simply managing symptoms and start tackling the underlying causes of these challenges.
It's a paradigm shift, really. And those medications you mentioned, their potential benefits might extend far beyond the realm of space travel.
Hold on, are you suggesting that what we learned about muscle loss in space could help treat conditions here on Earth?
Absolutely. Remember those shared biological pathways we talked about?
Hmm.
The same processes that cause muscle atrophy and microgravity are also implicated in various muscle wasting diseases that affect millions of people worldwide.
It's almost like a cosmic boomerang effect. We delve into space research to protect astronauts, and the knowledge we gain circles back to improve healthcare here at home.
It's a testament to the interconnectedness of scientific exploration. By pushing the boundaries of our understanding in one area, we often stumble upon breakthroughs that have far-reaching implications in seemingly unrelated fields.
That's what makes this so exciting. It's not just about achieving milestones in space. It's about the potential to unlock solutions to some of humanity's most pressing challenges. But even with all these advancements, we've barely scratched the surface of what's possible, haven't we?
We're just at the beginning of this incredible journey of discovery. Every experiment, every observation, every tiny worm sent to space adds another piece to the puzzle.
This deep dive has certainly opened my eyes to the complexities of space biology and the incredible resilience of the human body.
It's been a privilege to share these insights with you.
And to you, our listener, we hope you've enjoyed this exploration into the fascinating world of microgravity and muscle atrophy. Who knows, maybe one day, thanks to the knowledge gained from these brave astronauts and those tiny C. elegans explorers, we'll conquer the challenges of long-duration space travel and unlock the secrets to healthier muscles both in space and here on Earth. Keep exploring, keep questioning, and never stop looking up at the stars.